Hi everyone. Today we're going to start off with today's study tip, which is reality check. And this is to make sure that you're really learning what you think you're learning and that you're retaining the information. So the first question is, are you studying every day? And are you studying for enough time? This is a 5-unit class, and it really takes studying every single day in order to do well. So even if you think you remember something from a previous class or you're really comfortable with some of the material, don't skip this easy stuff. Make sure you study it, but spend more of your time on the new or challenging concepts. Okay? And you can test your memory and understanding of all concepts from lecture. So it's not just the same to continue to listen to my lectures, follow along, or read the book. You need to be able to recall from memory under the pressure of an exam. So we need to find ways to test your memory and understanding of the material. You can use lecture slides as guides on what to read from the book. And you should try the book's practice questions too. Okay, so now we're going to get into chapter 7, talking about neurophysiology. This is going to be divided into 2 parts. It's a pretty long lecture, and it's going to cover the next 2 lectures worth of material. Okay, so chapter 7, we're in neurophysiology, learning about the nervous system. In the nervous system, our neurons look somewhat like this, and they do differ across the brain, across types of neurons, but the sort of classic neuron is this, where you have, here's your cell with a nucleus. This is a cell body surrounding it, and you'll note there's a bunch of projections coming off this neuron that are called dendrites. Those are the spots that connections from other cells are going to make with this cell. One of the long projections is called the axon. You can tell it's an axon because it's really long compared to the dendrites, and it's going to have these myelinated regions. So these regions where these cells, called Schwann cells, or oligodendrocytes if you're in the peripheral nervous system, are going to wrap around the axon. And these are cells that are full of a fatty tissue, okay? And when they wrap around this axon, it creates a myelin sheath. So you can see the different layers of myelin here. And we'll get to this later on, but it's really important that the myelinated regions and myelination in general is what allows our nervous system to work so quickly. Okay. There's unmyelinated regions between that are called nodes of Ranvier. Okay. When we think about the nervous system, we can classify it in different ways. Typically, it's classified as a central nervous system as compared to the peripheral nervous system. So the central nervous system is going to be the brain and spinal cord, and the peripheral is going to be everything outside of the brain and spinal cord. And when we look at these different neurons, we can see how they are bringing information to or from the central nervous system from the periphery. So let's imagine it's called a sensory neuron. Okay, sensory neuron is going to respond or fire when the receptors are activated, and the signal is going towards the CNS. In a motor neuron, something that controls a muscle, you'll note that the cell body is here in the CNS, and the signal travels from the CNS out to skeletal muscles. Autonomic motor neurons, things that are controlling smooth muscle, cardiac muscle, glands, have this also outward flow of information from the CNS to the PNS, but they tend to have ganglia in between. And ganglia are just groups of neurons that are outside the central nervous system. We talk about this as afferent, information coming into the central nervous system. Interneurons are ones that go connect between each other in the CNS, and information that travels out from the central nervous system out to the periphery is called efferent. So I always remember this as efferent, the E being exiting, okay? And afferent is going towards. Efferent is exiting the central nervous system, going from the central to the periphery. Okay. So we said afferent neurons, the ones that are going towards the CNS, are in the peripheral nervous system. And a sensory receptor generates an action potential. Remember, action potential is how our cells fire. The signal goes through an afferent fiber, the afferent axon, and the cell body is adjacent to the spinal cord. This kind of neuron ends at the spinal cord. For interneurons, these are entirely within the CNS. They connect afferent to efferent neurons. And efferent neurons, the ones that exit, are mostly in the peripheral nervous system, but the cell body's in the spinal cord, so the cell body's in the PNS or the CNS, and the signal is sent through the efferent fiber, so it's exiting the central nervous system. It sends information to effector organs. Okay. And just to think about some of the facts about the brain, the average number of neurons in the human brain is 100 billion. And they'll mention glia briefly. Glia are far more numerous in the brain than neurons, but so you've got a lot of cells in the brain, 100 billion neurons, and then 5 or 10 times as many glial cells in the brain as well. The rate of neuron growth during fetal development is around 250,000 neurons per minute are produced. So it's a huge time of brain development, certainly the fastest time of new neuron development in an organism's lifetime. Okay. So to think about how neurons function, we have to think back to that chapter 6, talked about the resting memory potential. Okay, so first I want you to think about what is the RMP and why is it what it is. Remember, so we have these cells, RMP, resting memory potential, at around -70. Why? So maybe you want to pause this video here and take a minute to write a paragraph about what this is. Okay, so here's our cell. We can actually take an electrode recording from our neuron, and we find cells at -70 at rest. If a cell increases membrane potential, that's called a depolarization. So a cell becomes less negative, it's depolarizing. Repolarization is returning back to rest. Hyperpolarization is membrane potential going down below resting potential. So when we think about this as a cell being more likely to fire as a depolarization, less likely to fire if you're hyperpolarizing. So these are going to be inhibitory signals. The signals that depolarize a neuron are going to be stimulatory signals. Okay, so what's happening here? Why is the RMP of neurons -70? So remember that all cells in the body are maintaining a potential difference in voltage across that membrane. And this difference in voltage is our resting memory potential. It's very commonly -70 in neurons. Okay. With its -70, this means that the inside of the cell is negatively charged in comparison to the outside of the cell. And large negative molecules are trapped inside the cell, but some positively charged ions that are tiny flow out of the cell. Remember, this is mostly potassium. Potassium is far more permeable to the cell than sodium is. What this results in then is an unequal distribution of ions between the inside and outside of the membrane. And what's maintaining that difference, partially or mostly, is that sodium-potassium pump. So remember, it's pumping 3 sodium out and 2 potassium in. Okay. So what I want you to do is go to your book and look at figure 7.11, so 7.11, because this figure shows the difference in potential in millivolts between an intracellular and extracellular recording, and it's displayed on a computer or an oscilloscope. Okay? The resting memory potential, the RMP of the axon, may be reduced, depolarized, or increased, hyperpolarized. So depolarization is seen when that line goes up, hyperpolarization when it goes down. Okay, so how do we get our cells to fire? How do we get our cells to create an action potential? All right, so we can look at this graph here on the bottom and all that on the next page, but our cell is here at -70 resting. Some kind of stimulus from other cells is going to open sodium channels. Remember that at rest, your cell has a high concentration of sodium outside the cell and a low concentration of sodium inside the cell. So when the stimulus opens sodium channels, sodium rushes into your cell. Okay? That's positive ion coming into the cell, which increases membrane potential, causes a depolarization. There's a point, usually around negative 55 millivolts. That's called threshold. So a cell has to get to that threshold, has to depolarize enough to get to that threshold in order to generate an action potential. So in this graph down here, we can see here's some failed initiations. So a stimulus comes, cell depolarizes a little bit, maybe a little bit more, but not enough to reach threshold. So that cell will not have an action potential. Only if the signal is strong enough that enough sodium comes in to get to threshold, then you see an action potential. Okay. When that cell reaches threshold, that causes voltage-sensitive sodium channels to open. So this voltage is a trigger. The cell reaches negative 55. That triggers a bunch of sodium channels to open. And now we have a huge influx of sodium. Massive depolarization. Lots of positive ion coming in. Big depolarization. Our cells typically reach somewhere around positive 30 millivolts when this happens. So it's a spike in membrane potential. Then, a little bit later, on a timing mechanism, the sodium channels close. Okay. Around the same time as the sodium channels closing, voltage-sensitive potassium channels open. Now, it's actually the same voltage that triggers the sodium channels to open, but the potassium channels are just slower. So they take a little bit longer to open. But when they do, potassium rushes out of the cell. Because remember, at rest, you have a high amount of potassium inside the cell and low amount outside. So when you open potassium channels, potassium leaves the cell. And this is repolarization because now you're losing positive ion. Your cell is becoming more negative, going back to that resting membrane potential. In fact, it repolarizes so much that it reaches negative 90 millivolts and you get a hyperpolarization. Okay, then the potassium channels close and our concentrations of sodium and potassium are restored by the pump. Those pumps are working all the time. Okay, and so they're restoring concentrations all the time, but especially after an action potential. I put a couple links up here for YouTube videos about action potentials you can watch. There's a lot of great examples of these online, and you can watch some more videos to get a better feel of what's happening here. Okay. Okay, so here's the whole thing. This is the very classic drawing of an action potential, where you have your cell here at rest. A stimulus increases this membrane potential to threshold because some sodium channels are open. So there's some positive ion coming into the cell. At threshold, those voltage-gated sodium channels open. You have massive depolarization because you have tons of sodium coming into your cell. And then around this point, you have potassium channels opening. And potassium is leaving the cell. You're losing positive ion, so it's coming back down and actually dips below resting. So it hyperpolarizes a bit and comes back to resting. During this time, when your cell is below resting potential, that's called the relative refractory period. And why it's relative is because you can get an action potential, but it's a lot harder than in your cells at rest. This part here is called the absolute refractory period because during action potential, you can't trigger another one. It has to go either be hyperpolarized or back to resting in order to trigger a second action potential. Okay, so in the nervous system, we think a lot about how these signals are encoded because you know that you're not just seeing the world in on and off switches, right? But that's kind of where we start from. We think about encoding information in the brain. And so how we perceive things like stimulus strength, how bright a light is or how loud a sound is, has to do with action potential frequency. So we can think about a medium loud sound makes your ear neurons fire some action potentials. A loud sound makes them fire a lot. And your brain then interprets that as strength. It says, oh, okay, our sound is kind of loud or really loud. Okay, so looking at how this signal, this action potential, moves down the axon, we again have to think about the sodium and potassium, right? Those are the two main ions that are moving during this potential that causes an effect on our neuron. So the action potential initiates, it's called the axon hillock, right here, where the axon connects to the cell body. It initiates there and then travels down the axon. Now, as it's traveling down the axon, what you really have is a tiny action potential here, and then next door here, and then next door here. And so you have those sodium channels opening, and then they open on the next piece, and then they open on the next piece of membrane. Okay? And this pushes this action potential then from the cell body down to the end of the axons, because they're all the axon terminals. Okay. So remember, I talked about absolute refractory periods and relative refractory periods. Here's showing you what a voltage-gated ion channel looks like. Okay. And when this channel is totally inactivated during the refractory period, you cannot open it again. You cannot get another action potential then, because these are physically closed and will not open again. Okay. So again, we have an absolute refractory period because those sodium channels are inactive. This means that you cannot trigger a second action potential during this time. The relative refractory period, you can. You just need a stronger stimulus because your cell is already hyperpolarized. Okay. So on that previous slide we showed you, the action potential can only go away from the cell body because of it initiates at that axon hillock and then moves down the axon on adjacent sections of that cell membrane. Okay. So this is a repeat of what we said before. During the first part of repolarization is your absolute refractory period. The later part is your relative refractory period. Okay, remember those myelin sheaths I mentioned before? Myelin sheaths are cells filled with a fatty tissue that are wrapping around your neurons. And so they create layers and layers and layers of these sheaths. And in the peripheral nervous system, it's called the oligodendrocytes. Okay, but same idea, they're wrapping around axons. And what happens then is not that the action potential actually depolarizes each individual section of membrane. That signal jumps. It's called saltatory conduction or jumping conduction, where the action potential, let's say we initiate one here, it jumps to the next node. So it's jumping from node of RON-VA to node of RON-VA along that neuron, which makes it happen way faster than in cells that don't have myelin. So we know this for a couple of reasons. One is that invertebrate animals don't have myelin sheaths and their neurons function a lot slower. And we know that there are certain diseases in humans that cause loss of myelination. And then we see things like slow movements, decreased speed of speech, and problems initiating movement in people that are losing myelin. Okay. So the Schwann cells are the ones that are in the peripheral nervous system. Oligodendrocytes are in the central nervous system. So they have the same function. They're both myelinating neurons. The Schwann cells are the ones that are doing it in the peripheral nervous system. Oligodendrocytes are the ones that are doing it in the central nervous system. And it's used to speed up conduction of impulses. So we don't see any ion movement across the myelin. We only see ion movement at nodes. So it's called saltatory conduction because it's jumping from node to node. Okay. So I'm going to take a break there, and you can come back for part 2 of neurophysiology.